Hello, everyone. My name is Vicky, a technical marketer at CJ Bio. Thank you for joining the CJ Tech Talk. Today, I would like to introduce GutLuk, which is GutHealth Solutions as a natural AGP alternative. Here are the contents of my presentation. Firstly, I will start with the introduction, followed by the development process. And then I will talk about how we verified GutLuk through in vitro and in vivo trials. Finally, I will conclude my presentation. First, I will explain the background of the product development. Do you know why controlling pathogens are so important to the livestock industry? The enteric diseases are considered one of the most critical problems affecting the economy and health in the industry. It is mostly caused by the overgrowth of opportunistic pathogens throughout the gastrointestinal tract. The figure below shows the list of the pathogens responsible for enteric disease throughout the entire production phases. Major pathogen group includes E. coli, C. perfringens, Salmonella species, and Leucaena species, and so on. So these are always major concerns to farmers and feed millers. So therefore, AGP antibiotic growth promoters has been used to control the overgrowth of opportunistic pathogens. Animal industry has been using prophylactic doses of antibiotics at a low level to promote the animal's growth. But if we overuse antibiotics, they develop resistance and become ineffective when we eventually need them for treatment. And this bacteria or superbugs can be transmitted to humans. That's the reason why the EU has banned the use of AGP since 2006. And the trend is expanding globally to the countries like Korea and Vietnam. To respond to the regulations on the prophylactic use of antibiotics, zinc oxide has been used to reduce post-weaning diarrhea. But the therapeutic use of zinc oxide was banned across the European countries because of the issues with the heavy metal emission in feces. Because of these regulations, we've been trying to find a smart alternative. As we already know, there are lots of AGP alternatives in the market, such as prebiotics, probiotics, and organic acids and bacteriophages. However, we wonder if these are really effect as antibiotics. So CJ Bio focused on the mode of action of AGP itself. While searching for the AGP alternatives, the scientists noticed that the use of AGP reduced the bile salt hydrolase activity. It is called bile salt hydrolase inhibition, BSHI for short. So this mechanism was found not only in antibiotics, but also in antimicrobial minerals like zinc oxide and copper sulfate. I guess you heard of bile acid, but you probably know bile acid as an emulsifier that helps lipid digestion. Other than the role as an emulsifier, bile salt also acts as an innate antibacterial agent. Bile salt can kill the orally introduced pathogenic bacteria by disrupting cell walls. In other words, unique and natural antibiotics already exist in our body, especially when bile acids are released into duodenum. Glycine or taurine are combined with the bile acid called conjugated bile acid or bile salt. The form of conjugated bile acid has strong bactericidal effect due to the increased solubility in the intestine. Here we confirm how strong the toxicity is with an in vitro test. We found that a higher concentration of conjugated bile acids can kill the bacteria like ETEC, Salmonella, and Clostridium. So I can say the bile acids are powerful measures to control the pathogen in the body. But the problem is bile salts gradually lose their antibacterial activity as they pass through the intestine because of the bile salt hydrolase, or BSH. This is BSH produced by commensal bacteria. The gut microbiota evolved to produce BSH to survive the bile salt toxicity as a defense mechanism. If bile salts are deactivated by BSH in the intestine, these cannot control the pathogen introduced from the environment outside, and commensal bacteria are hardly affected by salt as they developed detoxification mechanisms. And that's the reason why we need to control the BSH level. Once again, BSHI functionality is helping to maintain the antibiotic activity of biosalt. So CJ Bio developed a natural product with the BSHI functionality. The GutLuk is a natural product, so it is very safe without resistance and residue issues. By enhancing the biosalt functionality, GutLuk prevent the intestinal pathogen overgrowth. Followed by the introduction, I will explain the development process. Here, I would like to introduce how we selected the main ingredient. Firstly, more than 700 candidates were screened from 3 million papers with the keywords related to gut health. And then the selected candidates were tested for BSH and antioxidative, anti-inflammation capacity through in vitro tests. After all these tests, we finally narrowed down to the 3 key ingredients, which is tryptophan, epicatechins, and vitamin K. These are the combination of GutLuk. Epicatechin and vitamin K synergistically inhibit BSH to control pathogenic bacteria, while tryptophan promote the recovery of damaged intestinal epithelial cells. Just in case, if epicatechins are not familiar to you, epicatechins are flavonoid and known as strong antioxidants. It is naturally found in various plant-based food like green tea, cocoa, and fruits. Among those catechins, CJ Bio discovered the best combination of 4 epicatechin groups having strong BSHI functionality. We further look into synergy effect on BSHI. Surprisingly, the combination of epicatechins and vitamin K demonstrates significant BSHI efficacy. When you see the left of the slide, you can see the BSHI efficacy of each ingredient. With the combination of all epicatechins and vitamin K, it showed the same level as antibiotics. To see the clear synergy effect, the BSHA efficacy was tested in the lower concentration. Due to the synergy effect, the combination of vitamin K and epicatechins showed much higher efficacy than itself. GutLuk also has effect on anti-inflammation and antioxidation. We found that the combination of epicatechins and tryptophan reduced inflammation, which is indicated by reduction in TNF-alpha in treated sample. Also, the epicatechins and vitamin K together reduced oxidative stress. To summarize the overall process, we selected 3 key ingredients after years of research, and we confirmed that the combination of each ingredient has synergistic effect on BSH. And as the ingredients are phytogenic, additionally increases antioxidation and anti-inflammation capacity. With this final product of GutLuk, we compare the efficacy to the other product in the market. GutLuk showed a superior bactericidal effect compared with the competitor's product. Can you see the dark circles on the agar plate? The circles show the inhibition zone of bacteria. Firstly, we cultivated 3 pathogenic bacteria, including E. coli, C. perfringens, and Salmonella gallinarum. And then we dropped the testing product. If bacteria cannot survive because of the product, the surrounding area are turned to dark. As you can see, it turned out GutLuk has the highest level of bactericidal effect than other products. In the previous slide, I already explained that GutLuk has antioxidation and anti-inflammation effect. In this part, we compare those effects with the competitor's product. As you can see here, GutLuk showed a better effect on decreasing IL-8, which is an indicator for inflammation. In the right, you can see GutLuk showed a strong antioxidant effect, while the other product didn't show any effect at all. Going to the next page, I will introduce some of the piglet trials. Firstly, we verified the GutLuk at Dankook University in Korea. It was tested with the 192 piglets for 28 days. There are 6 groups of treatment. For the negative control, we use Basal diet without using any antibiotics and zinc oxide. For the positive controls, antibiotics and zinc oxide were added. For the rest of the groups, we added GutLuk with different dosages. Here are the results. First of all, GutLuk significantly improved ADG to the comparable level to the PC group. When it comes to intestinal health, firstly, we checked the BC ratio and diarrhea rate. Piglet fed the GutLuk diets improved gut integrity and diarrhea rate. Here, the BC ratio of GutLuk group is much higher than PC and NC groups. When you see the diarrhea rate, no diarrhea occurred with GutLuk. This trial was very meaningful for us because we could understand that the GutLuk actually manipulates the gut microbiota as we hypothesized. If you are not familiar with the beta diversity, it represents the difference between the ecosystem of each group. So it shows how similar or different bacteria species are between the treatment groups. So GutLuk shifted gut microbiota compared to the piglets fed PC and NC groups. We further look into what kind of bacteria has changed. It is found that the GutLuk increased beneficial bacteria and decreased opportunistic pathogens in phylum and genus levels. On the left, it shows the relative abundance in each group. The green color represents the Firmicutes phylum, and the violet is Bacteroidetes, and the orange represents the Proteobacteria. And you can see the Firmicutes are increased and Bacteroidetes were decreased by GutLuk. As you know, in the increased ratio of Firmicutes and Bacteroidetes show the positive effect on weight gain for pigs. Notably, the abundance of Proteobacteria was much reduced by GutLuk. As it is well known, the major pathogens like E. coli and Salmonella species belongs to Proteobacteria phylum. Then we zoomed into the genus level. It was realized that the GutLuk also boosted the abundance of Lactobacillus. So all these results prove the idea that the GutLuk maintains the antimicrobial activity of bile salt. So it helped to control the overgrowth of the pathogens while supporting the growth of beneficial bacteria. The second trial was conducted at Long Nam University in Vietnam with 144 piglets for 32 days. Here are the 4 treatment groups. For the negative control, we used Basal diet. For the positive group, the antibiotics and zinc oxide were added at the same time. For the rest of the groups, we added GutLuk with different dosages on the top of the NC diet. For the result, GutLuk improved ADG and FCR compared to both the NC diet and even the PC diet. Additionally, we found that AID of dry matter and crude protein and fat also increased with the GutLuk diet. And AID of crude fiber and minerals showed the same trend. It seems that the microbiota manipulation by GutLuk has resulted in an improvement in utilizing fiber sources. Today, I introduced only 2 trial data. Since we have more trial data, we can provide the details upon your request. Finally, I ended up with my summary of the talk. I would like to emphasize that GutLuk is different from AGPs and other natural alternatives. First, GutLuk keeps the balance of the gut microbiome community, and it doesn't remove the entire microbiome. And secondly, GutLuk is a resistance and residue-free product. As I said, it just maintains the innate antimicrobial system. Lastly, although GutLuk is a phytogenic product, it has superior antibacterial effect over competitors' products, which are verified through in vivo trials. Thank you for listening today, and if you have any questions, please contact your nearest CJ Bio.